It is about the most important technology that's going to affect our lives, CRISPR technology that will allow the editing of the human genome. And it's about the most important moral question you and your children will face, which is to what extent should we allow this technology to edit our human genome. Jennifer Doudna, who's with me, is the author of Crack in Creation, but more importantly, I guess, the author of a 2012, I think it was, scientific paper that basically explained how CRISPR, or CRISPR Cas9, is that how you say it? Yeah. Cas9, we'll call it CRISPR, can be used to edit the human genome. You can even now, before we get into it, start thinking of the implications of what that means, especially if we edit the the germline, that would allow it to be passed on to our children. But before we get to the ethical implications, I thought I'd start a little bit with the narrative of how you got there. I think on this stage, some of you remember we had Dr. James Watson, who first did the structure with Francis Crick of the double helix of DNA. And among the great things he did is, like Jennifer, he wrote a book about how he got there. And I think when you were 12 years old, your dad put that book by your bedside. So let's start there.
Right, okay. Well, first of all, good morning, everyone. It's a really great pleasure to be here, and certainly an honor to be here with you, Walter. Yeah, that story really was, for me, I think, the beginning of my interest in molecular science. My dad was a professor at the University of Hawaii, not in science. In fact, nobody in my family was a scientist. My father was somebody who liked to troll around in old used bookstores and things like that. He found this kind of dog-eared copy of the double helix, threw it on my bed, and when I read it, I realized that this was a story. It was kind of a detective novel in a way, but it was actually real life. It was real science. It was how you could figure out the structure of a molecule by doing investigative experiments. And from that moment on, I really thought that was the kind of thing I wanted to do in the future.
And there's a wonderful scene in there where Francis Crick wings into the Eagle Pub, I think is the way they describe it, and say, I have discovered the secret of life. Explain what he discovered.
Well, he discovered the structure.
You might want to pull that microphone up just a little.
Okay.
Yeah, right.
How's that? Maybe better.
Yeah.
Yeah, so he discovered the structure of the DNA double helix. So DNA is the code of life. It's the molecule that holds all the information in cells that tell cells how to grow and divide and become an organism or tissue or whatever. And they had discovered that it looks like literally two ribbons wrapped around each other, sort of a double helical structure. Why was that important? Well, it really mattered because it explained a lot of things about inheritance. It explained the way that information can be stored chemically in the cell and copied faithfully from generation to generation, because each strand of this double helix includes a set of letters of the DNA code that each pair with another letter on the other strand. And so it was a very beautiful way to explain a lot of questions that scientists had up until that point. It also, in many ways, I would say, ushered in the modern era of biology, because it opened the door to many of the kinds of technologies that we're now using, including CRISPR.
So you are a PhD at Harvard, went on to teach at Yale. You're now at Berkeley, where you run things. What you're famous for before CRISPR was understanding the structure of RNA, which is, I guess, the way DNA expresses itself in the human or any organism. Explain your RNA research, because that's before you even came to the notion of CRISPR, right?
Right. Well, I like to call RNA DNA's chemical cousin, and many people think that it actually came before DNA if we looked back far enough in evolution. It's a molecule that, unlike DNA, tends to exist in a single-stranded form, not a double helix, although it can form very complex three-dimensional shapes. And that was the question that I set out to address when I was a younger scientist, was what do those shapes look like in RNA? And again, why did we care? Well, it was an important question because, again, many people think that RNA was the early primordial molecule that could both store genetic information and replicate it. And my research as a younger scientist was to understand how that RNA replication might have actually been catalyzed by RNA molecules that could both store genetic information and replicate it and pass it on to new generations.
So what is the function of RNA that we know now?
Well, lots of things. So one of the fascinating things that's happened over the last two decades or so in biology is that we've appreciated that RNA is not, when I was learning biology originally, we sort of thought RNA was kind of a boring molecule. It was kind of the intermediary between DNA, which held the secret of life in a way, and protein molecules that conduct all the activities in cells. And we now understand that RNA molecules do lots and lots of things to control the way that genetic information is deployed in cells. And that's really what I've been interested in studying over my career, is how that kind of regulation works by RNA. And in fact, that was how we got into working on CRISPR.
But when you say DNA is expressed in cells, which means expressed in who we are, to what extent, what sort of things are determined by our DNA, and what sort of things are just guided by our DNA, but aren't completely determined by it?
That's the $64,000 question.
And we're not paying you that much, but you can attempt an answer.
Well, so, DNA, people have been trying to understand the code and what's in the DNA, what are all the genes that make up a human being, for example. And one of the great, I think it's great, things that's come out of that is that it's complicated, right? It's really complicated. It's not just a list of genes, but in fact, there are many layers to the way that that information is actually used. And I think this is what you're alluding to, is something that we call epigenetics, which sounds complicated. It really just means making chemical changes to DNA that don't alter the genes themselves, but change the way the information is actually used, the way
So
the
give me an
proteins are
example
made,
of one of our traits that's more epigenetic, meaning it's controlled partly by the environment and what we do, and what is like purely genetic and encoded.
Well, it's hard to give you a specific answer, but many people think that traits like things to do even with our personalities, how we interact with our environment, things that are more, it's very hard to put our finger on a particular gene that's responsible for intelligence, for example, that a lot of that is really a consequence not just of the genes in our DNA, but the way those genes are actually used, which is epigenetic.
But things like particular diseases or maybe even childhood obesity is more genetically determined.
That's what people think, yeah.
Yes.
Yep.
So you're doing RNA, and you get the structure of it, the atomic structure is pretty cool. And if I remember from the book correctly, another great woman, biochemist, gives you a phone call out of the blue, even though she's a colleague of yours, you don't know her, and she says to you, we're doing CRISPR, and we need to know how it relates to RNA. You want to be part of it.
Yeah, so that was actually Jill Banfield. So Jill is a colleague at Berkeley. She's a geobiologist, so she's not a biochemist. She works on bacteria and where they grow in the environment and how they behave and interact with viruses and things like that. Her research had uncovered a lot of examples of what we call CRISPR, which is an acronym that stands for a series of repeated sequences in DNA. So very easy to pick out. If you're reading the letters in the DNA code, you could see this repetitive sequence, very kind of unusual. And what was quite interesting about this pattern of sequences was that it included a series of unique sequences that were derived from viruses. And the question that Jill Banfield had that she was not equipped to answer in her own laboratory was whether those virally derived sequences stored within these CRISPR elements might in fact be copied into RNA molecules in bacteria and then used to protect the cells from viral infection.
They first discovered some of that, I think, in Spain, right? And
yes, yeah.
It was like in yogurt or something. They were.
Yeah, there were several microbiology labs that had very important early roles in it. So Francisco Mojica was in Spain. He was one of the people that coined the acronym CRISPR. And then there was a group at a yogurt company in Denmark, actually, originally, that was working on how to protect their yogurt cultures from viral infection and had uncovered CRISPRs and started harnessing them for the use in food preparation.
Now, just so the audience cannot feel any less smart than you, when you first heard it, you thought it was spelled CRISPR, as in with a CRISPER, and I think you looked it up and you realized, okay, there's no final E in it, and you decide, okay, I'm going to take on this question of CRISPR, right?
Well, it was just one of those things. It sounded so crazy to me that it seemed really interesting to try to pursue this. And I've always been, I think there's two kinds of scientists, right, sort of broadly speaking. There are those that dive extremely deeply into one area of biology and become the world's experts in it. And there are those who are more sort of at a smorgasbord, and they're picking things and looking at things and trying things. And I've always been more in the second category. And so when I heard about this, I just thought, well, that's so fascinating that I feel like I'd love to test, do some experiments and see whether it's really true.
How did you then get to the most amazing discovery of our time, which is CRISPR can edit our genetic code, our genomes?
Well, I think this is a great example of frankly, of small science and curiosity-driven research, as well as an international collaboration. All are things that have really characterized my career over the last 25 years. So I got together with a colleague, Emmanuelle Charpentier. We met at a conference. Neither of us knew each other beforehand. She was running a lab in Sweden at the time, and she was working in seemingly a very different area of science than me. She's a microbiologist, a medical microbiologist, studying bacteria that infect people. And one of those bacteria turned out to have a very interesting type of CRISPR system in which a single gene, a gene known as Cas9, seemed to be required for those cells to protect themselves from viruses using the CRISPR sequences. And the question was, how does this protein that's encoded by the Cas9 gene, how does it work? And she was not a biochemist, I was. We realized that we could get together and do some experiments to figure out the answer to this question. And the result of that collaboration was this publication in 2012 in which we described the fact that Cas9 is an amazing protein that can literally be programmed with little pieces of RNA that a scientist in the laboratory could easily change and sequence. And what it does is to use that piece of RNA that it holds onto to find a matching sequence of letters in a DNA molecule, for example, the DNA of a cell, a chromosome. And when it finds that matching sequence, it holds onto the DNA and makes a precise double-stranded break in the DNA.
In other words, it edits it as you would cut and paste.
It's like cutting and pasting. I like to use the analogy of word processing because it's very analogous to that. You think of the DNA code like the text of a document. This is the scissors that allows you to cut out text, change it. The cell takes over after the DNA is broken and makes a precise change at the site of the repair.
Now, I'm going to make a little detour here because the three major characters in this narrative so far, Jill, yourself, and Emmanuelle, are all women. And I think back to the double helix when they kind of ignored Rosalind Franklin, the only woman involved. Is this a change in science that, I don't think we've seen a major breakthroughs like this led this way? Or is this just coincidence?
I think it's interesting serendipity. I think women are certainly making more forays into the scientific world, as well as obviously in biotechnology and business. It's still harder, I would say, for women in my own experience, but I think that this is a great example where none of us planned it that way, but it just so happens that all of us were running research laboratories that were doing highly complementary kinds of work where it made it very easy for us to work together.
Why is it harder for women still?
Well, I think there's still biases. A lot of it is unintended bias, I would say, but just ways that women are excluded. I think women, if you've read the famous Sheryl Sandberg book, I think a lot of the things that she talks about in Lean In resonate with me and with my colleagues. I think women are a little more reluctant to step forward and volunteer for things, and they get volunteered for things that take them away from focusing on leadership roles and things like that. So I think it's a lot of subtle things.
Mm-hmm. When you got to the notion of editing a genetic sequence, or what is it you're editing exactly? I know it's a strand of DNA, but what would you call that length of strand that you edit?
I would call it a length of strand. I don't know.
Okay. But to some extent, it would have a gene function, right?
It might have a gene, or it might have a piece of sequence that controls a gene, right? So it could be either a gene itself or the part of the DNA that controls the gene. But yeah, I mean, you can make changes that are very precise down to, I mean, imagine this. Imagine being able to make a single change to a single letter in the 3 billion base pairs of the DNA of a human cell. That's now the kind of accuracy that we have with this technology.
So explain to me the scientific, and maybe we'll get to the moral difference of doing that in a human being in its cell, leave aside animals, which are perhaps easier, and doing it in a germline. What would it mean to do it in a germline?
Right, so if we talk about doing it in an adult, anything, adult person, plant, animal, we're talking about making changes to cells that in ways where those DNA changes are not heritable by future generations. But in the germline, that's a different scenario where now changes that are made to the DNA become part of the entire organism if those germ cells are allowed to develop into an animal, a whole organism. And those changes can be passed on to future generations. So it becomes a permanent alteration. And if you really think about it, it's really changing the evolution of that species at that point.
But our evolution has always changed,
Yeah,
right? So
right.
what's the difference here?
Here, I would say the difference is that we're doing it in a targeted fashion. We're making decisions consciously about, we're going to change this one gene or even a set of genes to do something that we think is desirable.
And the time scale is different.
And the time scale is very
Because it
short.
would take
Yeah.
millions of years. We can now do in about 20 minutes or so. Yeah.
Well, proverbially
in theory.
speaking. Yeah.
Start me with the animals. Give me a couple of examples, like baby pigs or whatever, that that science has already been able to use this to do.
Well, there's a lot of examples. So mice, that mice are used very commonly as models of human disease. So it's been possible to make mice that have changes to their DNA that make them more human-like in certain ways, make it easier to study the effects of therapeutics, drugs, for example, on genes. Similarly in, you mentioned pigs, so pigs, one of the attractive things with pigs right now is the idea of engineering them so they're better donors, organ donors for humans. And this is already being actively worked on both in research labs, but also in a startup company.
So you basically create pigs that become farms for organs for humans.
That's the idea. That's right.
And so what do you need? What happens to the pigs? What do you do? How do you change their genetic coding?
You can literally, program the DNA so that their organs or certain molecular properties that they have, their immune system, for example, looks more human-like. So you put actually transplanting genes or just making more subtle alterations to their DNA so that at a molecular level, they behave in a more human-like way.
And what about like mosquitoes that carry Zika or something? What could you do to help fix that?
Right, so this is another fascinating use of gene editing technology, the idea of gene drive. So this basically just means if you have a way to alter DNA that's very efficient, you can use it, you can set it up in a way that it will drive a genetic trait very quickly through a population, for example, a population of insects. And if one does this in mosquitoes, and this is already being worked on, in principle, one could create strains of mosquitoes that are resistant to viruses and thereby can't transmit Zika virus, dengue virus, et cetera.
But you could also create then just as easily mosquitoes that don't reproduce the same way, that say you cut back a population of mosquitoes.
Yeah, that's right.
And is that being done in response to the Zika virus? Are you using CRISPR technology yet to take on mosquitoes?
Well, I'm not doing it, but groups are doing it. So this is a very active area of research because I think many people imagine that this could be an effective way to control insects that would otherwise be, spreading disease.
And that passes along to mosquitoes from here on out, right? It's not just a specific mosquito, it's part of the germline of the mosquito species or whatever.
That's right, that's right.
But let's start a little on the moral thing there. When I was young, I read Rachel Carson, and we were able to get rid of mosquitoes. We did it with DDT, and a generation later, there were no pelicans in my home state of Louisiana almost. We didn't know the consequences of doing that. How do we know the consequences of what science is now doing to the mosquito population?
I would argue we don't, and I think that's where we have to be proceeding with real caution in something like that. I was at a talk recently, and somebody was talking about gene drive for mosquitoes, and they showed a slide of building a very large structure, sort of maybe the size of this tent, that is designed to contain these modified mosquitoes and really try to do experiments in a controlled environment to see what happens when you have a gene drive
Let's all
spreading.
picture what happens if you have a tent like this that's supposed to contain mosquitoes.
Well,
we have nutria in Louisiana. We had places that were supposed to contain them too.
yeah, it's a big challenge.
So who's in charge of saying stop?
Well, right now there are various, obviously, government regulatory agencies that are, in charge of controlling the, environmental release of organisms that are modified this way. But I would say that right now we're at an interesting time because, the thing about this technology is that it's moving incredibly fast. So just to give you a sense, this technology is really just barely 5 years old right now, right? And already, and we didn't talk about this yet, but it's already in clinical trials for cancer treatments in China. And it's sort of mind-boggling how the pace of just scientific research has picked up with this tool. I mean, I'm seeing now there literally probably a dozen at least, or maybe more, papers a week in the scientific literature using the CRISPR technology. So one of the big challenges is how do you keep government regulatory groups up to speed with this? How do you make sure they are aware of how fast things are moving, and the pace of government is not that fast?
I will give you an example of it from yesterday, which will either be reassuring or not. I guess I could say this. Tom Price, the Secretary of Health and Human Services, as you know, was here on the stage. And he's worried about the Affordable Care Act, but he's also, this is in his wheelhouse, something he should be thinking about. And he saw your book in my office, and he started asking about it. I said, actually, this will be even more important 50 years, 100 years from now, what you do on this, than what you do on the Affordable Care Act. It'll affect the world more. And he said, well, maybe I ought to read the book. So I gave him a copy. We'll see. You can send him a signed copy. So let's start talking about humans, if we may. Tell me, you've made I'm looking at the pictures, longer hair on sheep, virus-resistant pigs, hypoallergenic eggs. But then it gets to the part where you can actually start changing the human genome. Where will we start on that? What will we do first? I mean, the little blood diseases, cancers, what?
Yeah, so I think, the kinds of treatments that really are the focus right now of research are not, first of all, not in the germline, right? So we're really talking about what we call somatic cell changes to adults or kids, but not those that would become heritable. And it's like you said, I think it's very attractive to think of being able to cure diseases that have a known single mutation that's causative. So for example, sickle cell disease is one that's talked about a lot. It's attractive for a treatment like this because it's in the blood, so it's possible to take blood stem cells from a patient, do the editing outside the body, and then replace the correctly edited cells so they repopulate the blood supply. And the sickle cell mutation has been known for a long time. It's a severe disease that we have no treatments for it right now, and there's a fairly large group of people that are affected. So it's, I think that'll likely be one of the early targets of gene editing.
And as we do our little moral spectrum, That's pretty solidly in the, yeah, let's do that. It won't affect the germline, it won't affect children, but it will save people from a bad disease. In Guangzhou, because China's now ahead of us on this, so we'll get to the fact that we're not spending enough on research in this country, so China gets to take the lead. They're now using it for what?
Well, I think you might be referring to using it in embryos, right?
Correct.
Yeah. And they're actually, in China, have been working on asking the question, does this technology work in developing human embryos? Could we actually imagine someday using it to, maybe we want to correct the sickle cell mutation, but we want to do it not in someone who already has this disease as an adult, but we want to do it at the stage of embryogenesis. And so the first paper, and now there's several actually published, that was about this topic was published in the spring of 2015 using non-viable human embryos. And it really sparked, it attracted a huge amount of attention because I think it really brought to the forefront the idea that, this technology is really on our doorstep and we have to make a decision as a society. Are we going to proceed with this kind of
And when you
heritable
say the embryos, it meant that it would be all future generations would have this fix.
If those embryos were to be
Viable.
implanted, if they were viable and implanted, then in principle, yes.
And the fact that they were non-viable was just a small choice. They could have chosen to use viable ones.
Correct, yeah.
So
yeah.
this is ready to go.
Well,
I mean, in the next 5 to 10 years.
I certainly think in that period of time, yes. Yeah.
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 So what type of things if you were thinking of doing it, what would be the things that you would say, I want to apply it to this?
In embryos?
Yeah.
I personally am not ready to go there yet, I have to say. I think that, first of all, I think that there needs to be a broad, what we call sort of a broad societal consensus about whether that type of use of gene editing should proceed. And there obviously hasn't been the opportunity
So
yet
if you
for
knew
humans
that
to
somebody genetically, an embryo, was going to get a fatal blood disease, you would not fix it?
I would advise other approaches, I think, today. I think the use of it in a somatic cell kind of application should happen first. And partly for safety reasons, but really, frankly, also to give all of us, time to grapple with this issue. Are we going to start editing the germline? Because honestly, once that begins, I think it'll be very hard to stop it. It'll be very hard to say, I'm going to do this thing, but not that thing, because everybody's feelings about this, I think, will be different. And who decides? Who pays for it? There's a lot of.
But you say we, the responsible people, should pause, put a moratorium, not do it till we grapple. Your co-author, Sam Sternberg, right? I think was a graduate student of yours?
Yes, yeah.
And so a woman, I assume it's a woman by the pseudonym, named Christina, who's I assume, an entrepreneur type. This is very recently.
Yeah.
Comes to him and says, let's do it. And she's trying to commercialize this, and she would, I assume, make all of our children taller and smarter. And it's pretty easy to do. Let's take a specific example that you could do with the gene, which I think is have stronger bones. That's a pretty simple genetic thing, right?
Or bigger muscles.
Bigger muscles. Two things that people might want to say, I want all my kids and kids' kids to have stronger bones and bigger muscles. And that's scientifically conceivable because those are truly things you can find on the genome that you could change, right?
Right.
I want to make sure I have the science right.
Yeah.
So Christina goes to your partner and says, let's market this. What happened?
That's and by the way, that's a true story.
And you won't give us her last name, right?
That blew my mind. Oh, do you want her last name?
Yeah.
We'll talk later.
Just tell the camera. I actually think I'm not going to go there, but this is something that ought to be talked about more. If there are Silicon Valley entrepreneurs trying to hire you and your graduate students to market this to make people's kids have stronger bones or bigger muscles, there probably should be more publicity on
To
it.
my knowledge, that isn't happening today, but that doesn't mean it won't in the future. I certainly am aware
But wait,
of
what
people
happened?
that
She
are...
came to Sam, right?
Well, this person really came to my student, yes, and said that she said she herself wanted to have the first CRISPR baby, and that she wanted to commercialize the technology, create a company that would offer this service to parents and allow them a, kind of a menu of options. And wow, we were really pretty shocked at the time, Sam and I, not so much now given all that's gone on, but I think it really does illustrate a couple of important points. I think you're bringing up this idea that there's a whole commercial aspect to all of this that is something that I think people are, we're all sort of grappling with. And secondly, it really does get to the moral and ethical challenges around this technology. Christina could not do that today in the United States, right? It would not be possible for her to do that, but could she do it in certain other parts of the world? Potentially, yes.
Will you suspect that Christina, or some Christina like that, is now in Guangzhou, China, trying to make that deal?
Let's just say I wouldn't be surprised if I were told that were true.
So people in the United States who are wealthy enough and have the feeling they could go this far in the moral spectrum could, in theory, go to a company in the foreseeable future, say 5 to 10 years, and say, here's the menu I want on my baby.
I think it could happen, yes.
And let's start thinking now through the moral things. Suppose in the genetic line, a family, they've got a blood disease, whatever it may be. Would that be okay to say, let's turn that one off?
Are you asking me my personal opinion?
Yeah.
Or?
I mean, we could ask. Yeah.
Well, again, I think it comes down to, to me, I think with any technology, you first and foremost have to ask sort of risk versus benefit. So if it were me, I'd want to know, first of all, does this even work, right? Is this company, do they have any credentials? Do they have any evidence? What's the safety of this? Does it work? And then you have to decide, is the risk, because there's always risk, is the risk worth the benefit? Are there alternatives that would be better or just as good that I should consider? I think we have to do that with any therapy or technology.
Suppose the benefit is better than the risk.
Well, then I think at some point it might be something we have to consider. I mean, we had an interesting meeting in early January of 2015. I think we talk about it in the book, where a group of scientists, was kind of a smaller group, about 20 people, including Paul Berg and David Baltimore, who had been involved in the early discussions in the 1970s around the ethics of molecular cloning. And
why don't let's pause there, because there's something famous called the Berg letter on cloning, where they say moratorium on cloning.
Right.
And that's kind of held, right?
Well, we're talking about two different things. So, molecular cloning means making copies of little pieces of DNA in bacteria, and that has been shown to be quite safe to do. So, so that's done widely now across the world in biology labs.
So flash forward to today.
Yeah.
Could you all have a moratorium because all of you scientists get together, or is that wishful thinking?
Well, I think you could. I mean, I think, that was the idea of that early meeting, was to ask, would it be possible to build a consensus globally among the scientific and clinical communities about the way to proceed with this very powerful technology? And that's really what many people are now working to do. But the point I wanted to make about that meeting was I thought it was very interesting that even in that small group of scientists, who one could argue are in some ways, all cut from the same cloth in a way, right? But we were having this conversation, and it was quite a heated conversation. And at one point, somebody leaned across the table and said, at some point we may decide it's not ethical to not use this in the germline.
Well, that's what I'm asking.
For certain things. And it kind of made everybody sit back and think about it a little bit differently. So I think there's still a lot of work to be done to develop the technology to the point where it would be, in principle, even safe enough to do that, in my opinion.
Yeah, but I mean, that's 5 years from now or so.
Yeah.
So we might as well start the moral thought now.
Exactly, we have
to. So the question is, as I asked you earlier, but you asked at that meeting, wouldn't that be immoral to say to a family, your kid has this genetic, easily marked trait that's going to blood disorder, not to fix it, right?
If there was no other treatment and the treatment
Okay,
was
but
shown
let's
to
go
be
down the
relatively safe,
spectrum. Suppose the kid is going to be born deaf. Would you fix that?
that gets into a very interesting realm because I've had a number of conversations with people in that community. Many of them actually feel that, that deafness for them is not a defect that they would fix.
So suppose you had two parents, both deaf, and from genetic reasons, and they felt it was not a defect, and they were about to have a child that was not deaf. Could they ask to fix it so their child would be deaf?
Whoa.
By the way, Michael Sandel asked that question in his class. I didn't.
Well, right. I mean, this gets into the realm of who decides, right? Who decides? Should the parents decide? Should they be told they can't do that? If they want to do it, should they be told they can do it, but only if they want to pay for it? I think these are tricky issues.
But if it's going to go down the germline, it's not their just their decision. And their child doesn't have a voice, but one has to think of the interests of the child too, right?
Yeah.
So as you go through the spectrum of things you could do, certainly, as we said, bone mass, muscle, perhaps even height, to some extent other traits. Where is there some moral line, or is this just some big old slippery slope?
I think that's the question, right? It's really that we're all grappling with. Where is there a line, and if there is, where is it? I think it's hard, honestly, to, you look at what's happened in vitro fertilization over the last, couple of decades. I mean, I'm old enough to remember before and after, right? And, there was a lot of controversy when in vitro fertilization first became available, right? A lot of people said, well, that seems wrong somehow. I remember my own parents saying, that seems wrong, test tube babies, that seems really wrong. And then, as there was obviously demand for it from infertile couples, and it was shown over time to be apparently safe to do, it's become accepted, at least largely. And now, if you go to different in vitro fertilization clinics in different states, they offer different things. So some will offer the possibility for parents to select the sex of their child, some don't, right? So some do and some don't. It's a very funny thing, and there's the regulation around this is a bit nebulous. So will that happen also with gene editing? It could, I don't know.
When you say you're trying to pull together a consensus on it, did you bring in the Chinese researchers from the Guangzhou lab?
We did, absolutely.
And what did they say?
Well, they were very interesting. I mean, they acknowledged the controversy around the work they were doing, but they were very frank, very honest. They said, look, in our society, in our culture, there's a very different view about human life and about early embryos than sort of the Western Christian Judeo tradition. So it's just it's a different culture, and I think it's just something that we have to grapple with.
I'm going to quote a sentence from your book. The argument that germline editing is somehow unnatural doesn't carry much weight with me anymore. What happened? Go ahead.
Yeah, well, I described this in the book. I really found, this was a surprise to me, actually, but I found my own attitudes about editing the germline changing over time because, for many reasons. I guess I started thinking about the fact that, after all, we pick our partners and we have kids, so that at some level we're affecting our kids just by our choice of partner. Actually, these days, you can, believe it or not, you can buy eggs, right? You can buy eggs, you can go to a sperm bank. I mean, you can look in a catalog and decide who do I want the father of my child to be from a sperm bank if you want to. That's already being done. And, and then, there are countries like Israel that actually pay, they pay for couples to have up to 2 kids by in vitro fertilization if they want to, and they pay for pre-implantation genetic diagnosis to remove embryos that have devastating genetic diseases associated with them. So there's already a lot of engineering in a way that's going on, right?
Yeah, so, Germany did that in the 1930s.
Well, exactly. So it's not a straightforward thing at all, But But the fact is that it does go on, and I think that, and I also, the other thing that happened was that, here I am a biochemist, and I do a lot of just, I've always done very fundamental research on molecules. I don't do anything with embryos or even with animals in my laboratory, and yet I was getting contacted, and this happens now routinely, by patients, families, parents who reach out and say, I have this disease in my family. A lot of them send pictures of their children, very beautiful children, and they're facing a devastating disease. And that hits you very, very deeply. And you start to ask, well, if this technology were available in a way that prevented that kind of suffering, why would we not want to use it?
And so can we draw not a sharp line, but try to put a line in the sand between fixing things that are diseases, very harmful to people, versus creating enhancements, like making children taller, muscles, smarter, blonder. People say, okay, I want to change race. I want my children to be a different race. That's not a disease. That's something. Can we draw a line between that type of thing and saying, I've got a genetic disorder that's going to destroy my blood, or is there no line to be drawn?
I think it's hard, and the reason is this. Like, let's say that I told you we can make a change to an embryo that will remove a single gene that, if left in place, will make a person susceptible to cardiovascular disease when they get older. And there's no deleterious effect of removing this gene, so it's a good idea to do it.
And
would you call that
you could do a bad
enhancement?
cholesterol already, right? I mean, you could do CRISPR to take out,
Exactly.
not Mike, but somebody's bad cholesterol.
That's
The whole germline.
That's the idea.
And that's right, as you're saying, that's right on the borderline of a
Would you
disease
call it
enhancement.
enhancement, or would you call it preventing disease? I don't know. It's a little of both, right?
And is there any way anybody's going to decide that, or is it going to be a global free-for-all?
Boy, that's a great question. I think in the end, it may end up being regulated differently in different jurisdictions, probably. I suspect, just because people's opinions and values are going to be different, and it's hard to change that.
When we get to the borderline thing, one of the things that amused me is you can get rid of armpit odor easily with CRISPR. Is that something we should do here, Matt?
Could be very useful.
One of the things in a broader sense is that, talking to Secretary Price, but also others, people question the value of basic science. They want to get rid of NIH and, National Science Foundation. And yet, it seems to me that everything, from the sequencing of the human genome to the ability to etch transistors on a piece of semiconducting material, all comes out of pure basic research labs like yours. What would happen to a lab like yours, which is really just a wonderful group of people, graduate students, people doing experiments, if the government quit funding basic research?
It would be a disaster. I mean, we would probably just mostly fold up our operation and go do something else. I mean, this is the thing. I think, we've been facing this in the US for the last decade at least, this push towards, initially it was really this push towards what's called translational research. In other words, people saying, why are we working on fruit flies and, fungi when really what we want to be doing is curing cancer and, curing Alzheimer's? Well, I don't think anybody would argue that, of course, we want to deal with cancer and Alzheimer's and other diseases, but the question is, how do you get there? And what's happened, if you look back over sort of the history of modern medical science, a lot of the fundamental discoveries and the technologies that enable those discoveries have come about through curiosity-driven research, projects that are not aimed in any particular direction. They're just a group of smart people, often a small group of smart people that are just asking, gee, I wonder how this works. And they do experiments, and that was very true with this whole CRISPR project for us, that lead in a very unexpected direction. I think that, that there has to be a balance. It's not to say that we don't want to have people working in targeted ways on diseases. We need that too, but we need both. And the danger right now, I think, is that, and you alluded to this earlier, right, is that if the United States really cuts back on funding for that kind of fundamental curiosity-driven research, a lot of it done in small, laboratories, I think we're going to find ourselves falling behind other countries. And already we're on the cusp of that happening because countries like China are investing huge amounts of money in that kind of
China,
research.
I once read, is investing 20 times more than the US in basic research in genetic technology. Does that sound right?
That sounds about right. My own colleagues, we struggle here in the United States to get, and I'm at Berkeley, I'm at one of the top research universities, but we struggle to get enough money, to put together money to buy equipment like electron microscopes, another field that's gone through this huge, explosion over the last few years in the advances of the technology. And meanwhile, we see our colleagues in China buying up, 20 at a time. And it's really astounding.
And you needed that microscope to figure out, say, the molecular and atomic structure of RNA.
That's absolutely right.
So I could do the thought experiment, which is suppose we had done this 70 years ago and stopped, as Vannevar Bush pushed basic research. We had not done it if Eisenhower had not done it. And we hadn't invented the transistor, had not invented the microchip, had not invented the laser, had not invented the internet, and not been able to do circuits and GPS, that sort of thing. That's what would have happened if we hadn't done the basic research on semiconducting materials, various things. And someplace like Russia or China had actually invented everything, from the microchip to the internet to the personal computer to GPS and phones. You could imagine Russia being the dominant economy in the world, right? So can you imagine China being the dominant economy in the world if we cut back basic research?
Sure. Yeah, I think we all wonder about it in the scientific community for sure. We joke about, someday we'll all be working in somewhere in China, running a lab there if we're lucky.
Not a very funny joke.
But it's a very real question, I think, for many of us is, what is the future of scientific research in this country? Are we going to maintain our predominance in that area, or are we going to let it slip away?
Now, this will be the last question I'm going to ask, so think of what you want to do with your grandchildren's genome or germline, and I'll let the audience go. A lot of research in this field is very collaborative, and then it's also competitive, almost like any other, whether it's Amazon and Google or whatever, there's competition and collaboration. But in science, there's certain things that tend to, it seems to me, but I want you to push back if I'm wrong, promote a little bit more competition than they do collaboration. For example, in your field, there's been some controversy where you have George Church and others at Harvard who've done things in their lab, Eric Lander at the Broad, and I can't remember.
And Feng Zhang.
Feng Zhang,
yeah.
is that pronounced?
Right.
He's done a lot on CRISPR. You all have been even battling over patents that deal with it. Eric wrote a piece called The Heroes of CRISPR that got a lot of criticism because it minimized your role, and he was hit both for being ungenerous scientifically and also perhaps sexist. I think he got hit for it too. But part of this competition seems to me to be driven by two things. One, a patent office that needs to find something that is hard to find in science these days, like who gets the credit for this amazing thing? And secondly, a Nobel Prize committee that can only award it to 3 people. Does this bother you? Is this a problem in science now?
I think it is a problem, definitely. I don't know how one solves this problem. I think, the truth is, I think that, science always has included elements of both collaboration and competition. And you need both in a way. Competition can be very good. It obviously spurs people on to do
Read the double
good
helix.
work.
It was, Linus
Exactly.
Pauling, Wilkins versus.
Yeah, kind of a race, and
yeah.
And the challenge is how to get that balance right. And I think one of the things that I think about a lot is how to attract younger scientists into our field. I think we really want to draw in, because they're the ones, honestly, that are driving the work right now. Am I in my lab actually pipetting? No, I'm sitting here talking to you, right? So, but people in my lab are there doing it, and they're the ones that are really driving the next results that'll be coming out. And so how do we ensure that they continue to be attracted to our field and drawn into it? And I think that, one runs, if there's a danger of, especially certain types of people feeling excluded, if there's a feeling of unfairness somehow, that can be very detrimental to attracting younger scientists. And same thing with prizes. I think the problem with prizes is that it's very difficult, and I now sit on various prize committees, as you can imagine. And so just thinking about when you want to give a prize in a certain area, you want to recognize scientists that have done the work, but you appreciate that, at some level, everybody's work is built on other people's and involves the work of a lot of younger scientists in the laboratories who aren't being sort of named in particular by these prizes. So how do you deal with that? It's very.
And so how much would you say you had to depend on, even though you're competing against sort of the George Church or the Broad Institute and others, and how would that be made better? Because, in your book you don't talk about them, in his article he doesn't talk about you, and it feels to me that, if I may, I could tell a story about what this happened in technology with the microchip, where both Texas Instruments with Jack Kilby and Bob Noyce at Intel co-invent in around the same time. And it's a 20-year patent battle and a Nobel Prize battle or whatever. But finally, Noyce calls Kilby and says, look, let's just share the patent. And they do. And when Kilby gets the Nobel Prize, because Noyce had died, he said, if Noyce were alive, he'd be standing with me here. Would you like to make a phone call and sort of bring all these people together at some point?
Well, it sounds lovely when you put it that way. Of course, life is always more complicated. First of all, I don't own any patents, and they're all owned by my university, and so it's my university that's making decisions about what to do with intellectual property. They are the ones hiring lawyers and deciding how to pursue things. And the same is true, I would imagine, at the Broad and MIT. So unfortunately, I think if it were up to the scientists, it might be better, but or maybe not.
We do have some lawyers in the room, so you don't have to keep criticizing and blaming them. But it would. Well, thank you. Let me open it up, if I may. Way in the back, first hand to be seen.
Dan Pearlman. I'm a scientist, and I've been following the CRISPR story. I'd like to have you imagine that we live in an age of 2017 where there is no ice cream. And you invent ice cream, and your friends come over, and you say, well, how do you like it? And they say, we love it. And then you say, well, it causes cholesterol elevation, and it may be dangerous, and it has some hazardous side effects. Maybe we shouldn't do anything about this. Well, maybe we should publish it on the internet. Well, I think CRISPR is sort of like that, because the cat is out of the hat, and capitalism is here to stay. And I don't understand, in what Walter alluded to, the free-for-all, how we'll have any control over it if patent offices are going to be irrelevant, et cetera. What's your scenario for the future, say over 5 to 10 years?
Okay, so you bring up a very good point, and that is that one of the things that's so on one hand wonderful, but also very challenging about this technology is that it's widely available, right? Whether patent offices notwithstanding, anybody doing academic or even commercial research right now can easily get a hold of the CRISPR molecules and tools for doing gene editing, and they can do it, and that's of course happening. So there's absolutely no way to put this back in the bag. I was at an ethics meeting recently, and a philosopher got up and said, if I could, I would throw away CRISPR. And I thought, well, but you can't. Yeah. So, it's sort of a moot point, because we can't do that. So I think you're right. I mean, it's forging ahead. We have to grapple with we're in the system we're in, right? And we have to deal with this technology in that context. So this is why I guess I've been encouraging an international discussion about it. I think the worst thing we could do is try to ignore it, or for scientists not to get involved in helping to educate people about it and help to make those decisions.
Yeah.
I have a question.
Yeah.
The negative and positive, as we know, chemotherapy came out of experimenting with germ warfare and mustard gas. So are we going to face gene warfare in the future?
Yeah, and you might even talk about, I guess it was James Clapper, the Director of National Intelligence. You were surprised to read it in the top 10 threat assessments to our country.
Yeah, so are we going to see gene warfare going on? Boy, I hope not, but I think it's something that has to be carefully considered. And yes, it was listed as one of the top weapons of mass destruction recently. And why is that? Well,
by the CIA and DNI.
Right. For these reasons, that it's widely available, it's accessible, it's a technology that's easy to use and easy to misuse. And so I don't have any easy answers to that, right? I think it's a big challenge that we face for sure.
Give me the nightmare scenario of a rogue regime or a non-state actor.
Well, you could certainly imagine misuse of this to, spread genetic traits in whatever, humans or something else that would have either deleterious effects or deleterious effects on other species. But honestly, do I worry about that more than I worry about other things, dangerous viruses that one can synthesize, toxins that are well known, how to make them. I mean, there's a lot of ways to do bad things, right? So I think this is another way to do bad things, but I'm not sure that it really is, that much different in a way than other technologies that one could misuse.
Yeah. Okay, there and then, well, actually, let's go to the way back because the microphone got to her first. And then the gentleman in the middle. Yeah.
Hi, my name is Maya Ishwaran, and I'm 18 years old. And I find your story very inspiring, especially for young women. And my question was, have you ever experienced discrimination based on your gender, and how do you deal with it in fields like science, technology, business, et cetera?
Well, when I was in high school, my high school guidance counselor asked me what did I want to do when I grew up, and I said, well, I want to be a scientist. And he laughed, and he said, girls don't do science. So maybe that's...
Where is he now? Can we send him the link to the video?
Sure, we can send him the link. So that was probably the only really overt time that I remember, something like that happening. I think that, I have to say that I have really been fortunate in my career, and I'm really grateful to many people that have supported me, helped me out, men and women, who have, given me encouragement at moments when I thought about quitting, which, happens has happened many times. So I have to say I've been really fortunate that way. But I talk to a lot of women who definitely do feel held back in various ways. And again, in my experience, in many cases, it's kind of unintentional discrimination, right? It's not sort of actively trying to prevent women from moving ahead. It's just not doing things that would be beneficial to them in different ways, whether it's at earlier stages, encouraging them to go into the STEM fields, stick with the STEM fields, or whether it's at the level of becoming a professor or a CEO of a company and needing things like childcare and to arrange your schedule so that it accommodates your personal life as well. So I just think there's still more work to be done in that area, and we have to do it at your generation, frankly, right? Just really kind of encourage people to think about enabling women in ways that will be effective for them.
Right here, Todd, yeah. And then I'll go the way back there, yeah. Yeah, yell and scream.
So there have been so many medical advances over the last 50 to 100 years, and this one is stunning, but is it different? And how is it really?
Why is this one different?
Yeah, well...
Huh? The question is, why is this one different from all the other medical advances?
Over the last 50 or 100 years, is this different?
Is this a difference?
And if so, why is it different? Yeah, and I think it is different. It's first of all, it's enabling in a way that most things are not. And it's enabling not just in medical science and clinical applications, but for everything. So we didn't talk too much about this, but it's also being very widely used now in agriculture, for animal husbandry, for synthetic biology. It's just really anything you can think of that has to do with biology. This technology touches upon. So it's very widely, impactful in that sense. And it's also relatively straightforward to use. So a crazy example of that is, Indiegogo now sells a kit you can buy for kids. Hey kids, use CRISPR, make your own CRISPR-edited a bacteria. And this is for
This is not a good
grade
idea.
school kids. So, you know,
Bad idea. Yes, I think standing up. I have trouble, there's lights in my eyes, so forgive me.
No problem. Peace, everyone. My name is Jada Imani. I'm a recent high school graduate, 2017, from Berkeley High, actually. Hey, and so to give context for the question, one, it's a context in what you were talking about with you wanting young scientists to be like interested and excited about this, but there might be concerns about fairness and things like that. That really resonated with me. Secondly, I'm part of a community that's been historically oppressed through the idea of genetic inferiority.
Yeah.
And still to this day, there's many systematic discriminations that we face. And so my question is, how do we make sure, and is there any way that you can encourage us to allow our compassionate evolution and our evolution of our moral conscience to evolve at the same rate as our technology?
This is the best question of the day.
it's a great question.
And it's great because it'll be the final question. But I know in your book you talk about making it equitable, making it fair, making it so that it's non-discriminatory. So why don't you end with that?
Yeah. Well, I guess you know, it is a great question, and it's something I think about a lot. And I've actually been working with a group at Harvard Medical School run by a wonderful professor, Ting Wu, and it's called Personal Genetics Education, PGEd. I encourage anyone who's interested to look it up, pged.org. And it's a nonprofit, and what they do is they actually outreach to groups that have been traditionally excluded from genetics, you know, understanding, you know, what is genetics all about? How does it affect me personally? And I think what she's doing is very important because her mission is to be inclusive. I think the only way we can proceed is really to have, you know, an open community of people where we invite everyone to get involved. It can't be just the elites doing something and everybody else, you know, sort of trying to figure it out and figure out how it's affecting them. I think it really has to be a societal and global effort. And I think PGEd is doing a great job of trying to do that outreach.
In the history of science, there's been almost no examples of advances in technology and science outrunning our moral processing power to deal with them. Could argue maybe the atom bomb, but there are few examples. This, to me, is one of the closest calls we're going to have in our lifetimes, and I'm really glad that you're part of the discussion. Thank you.
Thank you, Walter.